Welcome to the Longevity Papers podcast. This week we're diving into a, really fascinating collection of recently published research that's truly moving the needle in longevity science. We've got papers covering everything from, the fundamental genetic trade-offs that influence aging to critical advances in cellular reprogramming and even some novel ways to measure biological age.
Indeed. And our mission today really is to unpack the most impactful findings from this past week. We want to help you, our listener, quickly grasp not just what was found, but maybe more importantly, why it matters for the field, particularly for biotech, and what crucial next steps these discoveries might suggest. We'll do a full deep dive into two particularly significant papers, and then we'll give you a concise overview of three others you should definitely be aware of.
Okay, sounds great. So let's jump right in and kick off our exploration of the latest breakthroughs. Right, first up, our first deep dive. This is on a paper that's, it's already generated a lot of buzz, definitely a must-read. It was published just yesterday, June 26, 2025, in bioRxiv. That's pronounced bioRxiv, for those maybe unfamiliar. It's a preprint server. The paper is titled, An Antagonistically Pleiotropic Gene Regulates Vertebrate Growth, Maturity, and Aging. The authors are Moses, Franek, Atlan, and colleagues from places like the Hebrew University of Jerusalem, University of South Bohemia, and University of East Anglia.
Yeah, this study is highly significant. I mean, it offers some of the most direct evidence we've seen to date for a really foundational concept in aging biology, the antagonistic pleiotropy theory. For decades, this theory has been around the idea that genes which are beneficial early in life can unfortunately have detrimental effects later on. But finding direct single-gene evidence for this, especially in a vertebrate, that's been quite scarce. So this work really moves us from a somewhat abstract concept to a concrete, maybe even potentially druggable pathway, which is, very exciting for the field.
That is quite a leap. Okay, let's unpack this a bit then.
Yeah.
They identified this specific gene, VGL-L3, in the turquoise killifish Nothobranchius verseri. What exactly is this gene doing? How does it create that trade-off?
Well, what's fascinating here is how clearly they demonstrate this dual role. They show VGL3 expression is concentrated in male reproductive cells, Sertoli, and Leydig cells in the killifish gonad. So that hints very strongly at a reproductive role. And they also note its gene architecture, especially a shorter version, or isoform, is remarkably conserved across a huge range of vertebrates. We're talking fish all the way to lions. That deep conservation really suggests it's fundamentally important. The researchers then used CRISPR, the gene editing tool, to create very specific mutations in these VGL3 isoforms so they could study the effects.
Right, CRISPR. So when they made those changes, what was the benefit? They found an acceleration of maturity in the mutant males, is that right?
Precisely. The mutant males, specifically the ones with this VGL3 X1 mutation, they showed accelerated nuptial coloration, the bright breeding colors at just 1 month old. That means earlier sexual maturity. And this was accompanied by increased body weight and, importantly, a significantly higher gonadosomatic index. That basically indicates a greater investment in their gonads, their reproductive organs, early in life.
Okay, so that's the early life benefit. What about the late life cost then? Where does the trade-off bite?
The trade-off becomes starkly clear when these fish get older. These very same VGL3 EGUS1 mutant males, they developed a massively increased incidence of large black melanocyte expansions on their tails, and these look very much like melanoma-like tumors.
Yeah.
Now, to definitively prove these were cancerous, the authors did something really quite innovative. They first had to create a totally novel tool, an immunodeficient killifish, a RAG2 knockout mutant, basically a fish engineered without a functional immune system.
Wow, okay, that sounds like a huge methodological advance in itself. So how did creating that immunodeficient fish help them prove the cancer link?
Well, this new tool enabled the absolutely crucial experiment. They transplanted cells from one of those VGL-free mutants' melanocytic expansions directly into this new RAG2 mutant fish. And what happened, those transplanted cells formed large invasive tumors. They were actually invading muscle and blood vessels. And then histology and EDU staining confirmed these cells were highly proliferative, which is, characteristic of cancer.
Okay, so definitely cancerous. And what about the impact on overall lifespan? Did these fish die sooner?
It's interesting, the median lifespan shown in their Kaplan-Meier plot wasn't actually significantly different between the groups. But, and this is key, the age-specific mortality curve for those VGL3 EX1 EGA1 males showed a much steeper upward slope. This demonstrates an acceleration of what we call actuarial senescence. Basically, their risk of death increases much more rapidly with age, and that is the direct cost of their faster early life development.
Right. So a single gene in killifish directly trading early success for late life disease and a faster decline.
That's narrow.
That's powerful stuff. But, the skeptical question always comes up. How much can we really translate this from killifish to us to mammals?
That's the right question to ask. Look, it provides a definitive single-gene example of antagonistic pleiotropy in a vertebrate. That's a massive conceptual step forward. However, you're right, a skeptical but pragmatic view would absolutely highlight that this is, for now, a single study in one species of fish. The key experiment now, the next step for the field, is to see if modulating VGL3 or its downstream pathways in mammals can actually uncouple that early benefit from the late life cost. You could imagine experiments like generating conditional knockouts of VGL3 in mice, turning it off only later in life, perhaps, or maybe screening for small molecules that inhibit its specific activity, potentially as a late life only therapeutic. That's where the real biotech interest lies, I think.
Makes sense. Okay, let's move on to our second deep dive. This paper tackles, well, really one of the biggest bottlenecks in rejuvenation biotechnology. It's titled Comparative Analysis of Mouse Strains for In Vivo Induction of Reprogramming Factors, published June 25, 2025 in Cell Reports. The authors are Sarah Picot, Alba Vilches Acosta, João Agostinho de Sousa, and their colleagues, including Alejandro Ocampo, primarily from the University of Lausanne, with Epaterna Say and ETH Zurich involved too.
Yeah, this paper is absolutely crucial for the entire field. I really can't overstate that. In vivo reprogramming using those Yamanaka factors, OSKM, it holds immense promise, like true AIDS reversal potential. But the clinical path has been seriously blocked by major safety concerns, specifically forming teratomas, those mixed tissue tumors, and also the loss of essential cell identity in tissues. This paper directly addresses that bottleneck. It confronts those safety issues head on.
Right, so it's maybe not a flashy discovery of a totally new pathway, but potentially much more valuable in the long run, a foundational resource maybe.
Exactly. That's a great way to put it. By systematically creating and carefully characterizing new mouse models that are designed for safer induction, the authors are building the essential tools the field desperately needs to move forward. This is highly credible work, really solid, and it represents a critical, enabling step towards making systemic rejuvenation a potential reality.
Okay, now here's where it gets really interesting for me. Before they developed their new approaches, they compared existing mouse strains. And the results were pretty dramatic, weren't they?
Oh, they were. Very stark results. Their comparison of 4 existing strains showed that when you give continuous doxycycline, the drug used to switch on the factor strains like 4FJ and 4FK, where the reprogramming genes are stuck into the COL1A1 locus, well, they had a median survival of less than 10 days.
Less than 10 days. That's incredibly fast.
It is, and this correlated directly with extremely high levels of the OSKM transcripts, the reprogramming factor messages, in the liver and the small intestine. Those are the organs, of course, directly exposed to the doxycycline when it's put in the drinking water. And this really high uncontrolled expression led to a clear loss of cellular identity. You could see mature liver cell and gut cell gene expression signatures just plummeting. The cells are basically forgetting what they were supposed to be.
So uncontrolled high expression, especially in the liver and gut, seems to be a key driver of this toxicity. Okay, so what safer strategies did they manage to develop then?
This is where the clever engineering comes in. They devised several novel genetic strategies. One really successful approach involved engineering a mouse strain where Kindry recombinase a genetic switch specifically removes the OSKM genes just in the liver and intestine. Doing that dramatically extended survival from less than 2 weeks to over 40 days. That's powerful proof that these organs are a primary source of the systemic toxicity. And they had even more sophisticated strategies too, like using a Ki67 Cre driver. This removes the OSKM cassette mainly from actively dividing cells. That also significantly improves survival. And the thinking there is it probably preserves the gut lining, the epithelium, which constantly divides.
Interesting. And what about approaches that weren't about removing the factors, but maybe controlling them better, like chimeric models?
Right, they looked at that too. They created chimeric models using a CAG-Cre driver. This activates reprogramming, but only in a fraction of cells throughout the body, kind of a mosaic pattern. And this approach also significantly improves survival. When they looked at the transcripts, they saw it successfully reduced OSKM expression in those toxic hotspots, the liver and gut, while still allowing high expression in other places, like the kidney. So more targeted, less globally disruptive.
This really does feel like it provides an essential roadmap for the field. It shows that maybe toxicity can be uncoupled from the induction process itself.
It absolutely does. It's a huge step forward in that sense. These new mouse models are frankly invaluable tools. They'll allow researchers to start pursuing more potent, perhaps longer-term reprogramming protocols with a bit more confidence. Now, the skeptical view, of course, would be, are these models really safe enough yet for thinking about human translation? Or perhaps could the safest approaches also turn out to be the least effective at actually reversing age? That's a key tension. So we definitely need follow-up experiments, things like long-term lifespan studies, using these new mouse models under cyclic reprogramming, turning the factors on and off. We need to assess not just teratoma risk over the long haul, but also subtler issues, other pathologies, and the overall impact on health span and longevity. And crucially, we need head-to-head comparisons of these new models against older ones to see how well they actually reverse specific types of age-related damage in different tissues.
Lots of work still to do there. Okay, let's briefly turn our attention now to 3 other important papers that came out this week, starting with one that kind of challenges how we interpret a tool many people in geroscience use all the time.
Yes, this one's called Misalignment of Age Clocks, published June 25th in Geroscience. Authors are Xiaoyu Mei, Hanana Kabir, Michael Conboy, and their team at UC Berkeley. This is really essential housekeeping for the field, I'd say. It critically points out how most of the major epigenetic clocks we use are mathematically optimized purely for correlation with chronological age, and that comes at the expense of biological interpretability. They show these clocks are often incoherent, meaning they assign positive weights to both age increasing and age decreasing methylation changes, which doesn't make much biological sense. And they're also heavily skewed by leukocyte fractions, the miss of immune cells in your blood sample. This really demands we change how we interpret clock data. We need to be more critical.
Wow. So it's like realizing the speedometer in your car is really good at matching the speed limit signs, but maybe not reflecting your actual speed accurately under all conditions. A huge warning for researchers relying heavily on these biomarkers.
Exactly. A call for critical thinking about our tools. And speaking of clocks, here's a genuinely novel one. Epigenetic age prediction using N6-methyladenine in the bumblebee Bombus terrestris. This paper by Renard, Bozeret, and Aaron from Université Libre de Bruxelles was posted on Biocorp just yesterday, June 26th. This is potentially a paradigm shift. Why? Because all current epigenetic clocks, the ones the previous paper critiqued and others, they all rely on C5-methylcytosine or 5-MLC. The idea of building a clock from a completely different epigenetic mark, N6-methyladenine or 6NA, is just groundbreaking.
A whole new type of clock. And what did they find using the 6 millibaricol clock in bumblebees?
Well, they demonstrated 2 key things. First, that a 6-MLA-based clock can accurately predict chronological age in bumblebees. But maybe more importantly, it also seems to capture signals of biological aging. It predicted a younger age in bees whose lifespan was extended using a drug. And that is really the gold standard for a true aging biomarker reflecting biological rather than just chronological time. Now, the main skeptical point, obviously, is its relevance to mammals, because 6-MA seems to function quite differently in us compared to insects. But still, it opens up a whole new avenue, a new frontier in biomarker discovery.
Really fascinating. Okay, finally, let's touch on a paper that connects aging very directly to a real-world clinical problem that affects many people.
Right, this last one. Rejuvenation alleviates prolonged post-surgical pain in aging mice by mitigating inflammaging. Published June 23rd in the journal Pain. Authors are Basma Abdelkader, Xiangquan Shi, Wenbo Samzu, and colleagues from McGill University. What they did here was apply a classic concept, heterochronic parabiosis, basically sharing blood between a young and an old mouse, but they applied it to a novel and very clinically relevant problem: prolonged pain after surgery, which is more common in older individuals. It elegantly connects those systemic circulating factors associated with inflammaging, that low-grade chronic inflammation of aging, to a specific functional problem, pain sensitivity.
And the findings. What happened when they applied this old concept to this new problem?
The key finding was that giving serum from young mice to old mice could actually normalize their pain sensitivity and improve their recovery after a surgical procedure. This provides pretty strong evidence that factors circulating in the blood aren't just passive biomarkers of aging. They seem to be causal agents driving some age-related phenotypes, like increased pain. Their proteomic analysis also pointed towards some interesting sex-specific inflammatory patterns, which again highlights the need to move beyond only studying male mice in aging research. Now, the skeptical standpoint here is that serum is, a complex cocktail, hundreds, maybe thousands of things in there. This study doesn't pinpoint the specific factor or factors responsible for the pain relief, but even so, it's a really powerful proof of concept.
Absolutely. Wow, what an incredible week for longevity research when you line it all up like that. from pinpointing specific genes involved in those fundamental aging trade-offs to developing safer tools for something as futuristic as systemic rejuvenation, and then even unveiling completely new types of age biomarkers and finding new applications for old concepts like blood exchange. It's clear the field is just moving at an astonishing pace.
It really is. And these papers, taken together, highlight how our understanding of aging is becoming much more mechanistic, much more actionable. These aren't just observations anymore. They're providing concrete insights and, crucially, tools that could genuinely accelerate the translation of this research into real-world therapies. So for those of you listening who are in biotech, these aren't just, academic curiosities. These are foundational pieces for future development, for future innovation.
Absolutely. And that leads us perfectly to our final provocative thought for you, our listener. Given these rapid breakthroughs we're seeing in understanding and even starting to manipulate core aging mechanisms, what previously unimaginable clinical applications for longevity science might actually become commonplace within, say, the next decade? Things we might currently dismiss as science fiction. Something to ponder as you continue your own deep dives into the literature.
Thank you for joining us for this week's deep dive into the longevity papers.
We really hope this has given you a valuable head start on understanding the most important research from the past week. Until next time, keep digging into the data.